بسم اللہ الرحمن الرحیم۔ عزیز طلباء و طلبات، ہم میکرو مالیکیولز اور بایو مالیکیولز کو اسٹڈی کر رہے ہیں میڈیسنل کیمسٹری میں، اور اس میں 4 جو میکرو مالیکیولز ہیں وہ شامل ہیں۔ ایک آپ نے سب سے پہلے کاربوہائیڈریٹس کے بارے میں پڑھا جو کہ میکرو مالیکیول ہے اور بایو مالیکیول ہے۔ اس کے بعد آپ نے امینو ایسڈز اور پروٹینز کے بارے میں پڑھا کہ وہ بھی ایک بایو مالیکیولز ہیں اور میکرو مالیکیولز ہیں۔ تیسرا جو ہے وہ آپ نے لپڈز کے بارے میں پڑھا جو کہ بایو مالیکیولز اور میکرو مالیکیولز ہیں۔ اور چوتھے قسم کے جو میکرو مالیکیولز اور بایو مالیکیولز ہیں اس میں جو شامل ہیں وہ ہیں نیوکلیک ایسڈز۔ نیوکلیک ایسڈز نیوکلیک ایسڈز کیا ہوتے ہیں، کیسے آپ اس کو ڈیفائن کرتے ہیں، اور پھر نیوکلیک ایسڈز کی کون کون سی قسمیں ہیں، پھر نیوکلیک ایسڈز کے کیا کیا فنکشن ہیں، کیا یہ کام کرتے ہیں، اور پھر نیوکلیک ایسڈز کے کمپوزیشن کیا ہے، کن کن چیزوں سے یہ مل کے بنتے ہیں، اور پھر ان کی سٹرکچر جو ہے وہ نیوکلیک ایسڈ کی کیا ہے، یہ چیزیں انشاءاللہ آپ دیکھیں گے اس میں نیوکلیک ایسڈز میں۔ آج ہم نام تو آپ اس کا نیوکلیک ایسڈ کا سنتے رہے ہوں گے، یہ 8th 9th سے شروع ہوتا ہے، اس سے پہلے بھی بعض کچھ چیزوں میں سائنس کی بکس میں ملتا ہے۔ نیوکلیک ایسڈز کو ہم ڈیفائن اس طرح کر سکتے ہیں کہ نیوکلیک ایسڈز آر پالیمرز آف نیوکلیوٹائڈس۔ سمپلیسٹ ڈیفینیشن اس کی ہم اس طرح کر سکتے ہیں کہ نیوکلیک ایسڈز آر پالیمر آف نیوکلیوٹائڈس، یا ہم اس کو ایک اور طرح سے ڈیفائن کر سکتے ہیں نیوکلیک ایسڈز کو کہ نیوکلیک ایسڈز آر پالیمر آف دی نیوکلیوٹائڈس اینڈ the components of nucleic acids are number 1, 3 important components are present in the nucleic acids. Number 1, 3 types of the monomer units are present in the nucleic acid. Number 1 is heterocyclic aromatic amines bases derived from purine and pyrimidine. Nucleic acids are composed of the monomer units of heterocyclic aromatic amine bases derived from purine and pyrimidine. Number 1, this is one component. Number 2 is the monosaccharides, maybe D-ribose or 2-deoxy-D-ribose. And third component is the phosphoric acid. So, nucleic acids are polymer of the nucleotides, and nucleotides are composed of the monomer units of heterocyclic aromatic amine bases which are derived from purine and pyrimidine. We will see what is purine, what is pyrimidine, but for the moment you should keep it in your mind the definition of the nucleic acid. So nucleic acids are the polymer of the nucleotides, and which are composed of 3 important components. Number 1, the monomer units of the heterocyclic aromatic amine bases which are derived from purine and pyrimidine. And second component is the monomer units of the monosaccharides, which are the D-ribose or 2-deoxy-D-ribose. And third component is the phosphoric acid. So these three combine together and they form the nucleic acids. Now the function of the nucleic acid is what? The organization, maintenance and regulation of the cellular function, the function which are performed by the cell, require a tremendous amount of the information. Organization, maintenance and regulation of the cellular functions require tremendous amount of the information. Now where informations are stored, tremendous amount of the information are required, all of which must be passed on each time a cell is replicated. When a cell is divided, those information must be passed on from parent to offspring. This is called as this is the function. This is one of the important function of the nucleic acid. When DNA will be split up into two daughter or two new DNA, that is called as replication. Now the genetic informations are stored and transmitted from one generation to the next generation, from one generation to the next generation in the form of DNA. DNA contains chromosome, and in the chromosome there are present genes. Genes are the part of the DNA which contains codes for the characteristics, different characteristics, and genes are responsible for transmission of the characteristics or traits from parents to their offsprings or their children. So this is the function of the DNA. DNA has chromosomes and genes. In the genes, codes of for the characteristics and traits are present in the genes, and genes are responsible for the transmission of their characteristics of the parents or traits of the parents into their children. For example, a human being offspring or children will have the similar characteristic properties of the human, and monkeys offspring have characteristics of the monkeys, and the dog offspring has the characteristics of the dog. This is all due to the genes which are present in the nucleic acid. Genes are present, and these genes are responsible for the transmission of the characteristics of their parents into their children. Now, there is present another term which is called as transcription. Here one general term is the gene expression. Gene expression is consisting of two processes. One is called as transcription and other is called as translation. Copying of the informations of the DNA into RNA is called as transcription. Copying of information of DNA into RNA is called as transcription, and it takes place in the nucleus of the cell. What is the process? It is called as transcription. Copying of the information, copying of the genetic information of the DNA into RNA is called as transcription. And coded informations are used to produce protein is called as the gene expression. RNA produces proteins according to the information which are obtained or copied from the DNA. That is called as translation. Production of the proteins on the basis of information obtained from the DNA. It is production of the proteins by the RNA is called as translation. So DNA is copying of the information from the DNA into RNA, and then an RNA produces specific type of the proteins on the basis of information obtained from the DNA. That is called as translation. So transcription, replication, transcription and translations are involved in the nucleic acids. Now we see what type of here nucleic acids. Two types of the nucleic acids are present. One is called as DNA and another is called as RNA. For what DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid is for DNA and RNA is for ribonucleic acid. It stands for ribonucleic acid and DNA stands for deoxyribonucleic acid. So two types of the nucleic acids are present, and remember the definition. The nucleic acids are polymer of the nucleotides in which the purine or pyrimidine bases are present. Aromatic amine bases derived from the purine and pyrimidines are present, and ribose or deoxyribose is present, and phosphoric acids are present. Such polymers are called as nucleic acid. Nucleic acids are of two types: DNA, deoxyribonucleic acid, and ribonucleic acid. One is the difference between these two. Names of these two are different from one another. One is DNA, other is RNA. What other differences are present here? It is double-stranded, stranded. दो धागे वाले ये हैं DNA, alpha helix. This shape is called as alpha helix. DNA has two strands arranged in the alpha helix structure, and DNA stores genetic information, and just like that of in the library books are stored, it is a cellular library. In it genetic informations are stored, so it is called a cellular library. And DNA is found in the nucleus. It is located in the nucleus of the cell, and it is double stranded, strand, stranded. Two threads are present in a single DNA. Now we come to the RNA ribonucleic acid. How ribonucleic acid is different from the DNA? It is a single stranded, or a single thread is present. It is a single or single stranded RNA is present. Number two, DNA stores genetic information, while RNA transfers genetic informations, and on the basis of transfer of genetic information, it produces different type of the proteins. Here it is cellular library, and it synthesizes the proteins. DNA stores the information, RNA transfers the genetic information. It is cellular library-like function. It is synthesizing the protein. This is the second difference. Third difference is DNA is found in the nucleus, it is located in the nucleus, while RNA is mostly outside the nucleus of the cell. And the fourth difference is that it is double stranded, while RNA is a single stranded so these are the four important differences in the nucleic acid. And nucleic acids are these are 4 or 5 differences. Another difference is the nucleotides of the DNA and the nucleotides of the RNA are also different from one another. Here, this is a base derived from purine or pyrimidine. This is aromatic amine bases derived from purine or pyrimidine. We will see what are purine and pyrimidines. Then this component is called, represented as S as sugar. This is the deoxyribose. Here at 2 position H is present instead of OH. And another component is phosphate. So aromatic amines derived from purine and pyrimidine is present, and deoxyribose is present. Why it is deoxy at 2 position? Here you can see this. Here at 2 position OH was present, here 2 position only H is present. Here this is what? This is phosphorus, this is phosphorus. This is the number 1, 2, 3, 4 and 5 of the ribose. at 2 position, here H is present, here at 2 position, what is present? OH is present. So one difference of the DNA is it is deoxyribonucleic acid, its name is DNA. Another nucleic acid is RNA ribonucleic acid. Second difference, you have seen that DNA is found, it is stored inside the nucleus, and RNA is found outside the nucleus in the cell. Then you have seen that genetic informations are stored in the DNA, and transformation of the informations are through the RNA. Now this is another difference we have seen, that DNA is double-stranded, strand is double-stranded, while the RNA is single-stranded. Here another difference is that nucleotides of the DNA are different from the nucleotides of the RNA. We will see later on what is the nucleotides and what are the nucleosides. This is represented by B. It is a base, aromatic amine base, derived from purine or pyrimidine. And second component of the nucleotide is the sugar, 5 carbon sugar, which is the deoxy-D-ribose. At 2 position oxygen is not present, so deoxy-D-ribose is present. Second component of the nucleotide is the deoxy-D-ribose, and third component is the phosphoric acid or phosphate ion. So this is the structure of the nucleotides of the DNA. This is the structure of the nucleotides of the RNA. Here at 2 position OH is present, here at 2 position H is present. So this is the deoxy-D-ribose, this is D-ribose. What is this? This portion is representing the base derived from the purine or pyrimidine. This portion is representing the D-ribose. D-ribose. This portion is representing what? Phosphorus or phosphoric acid or phosphate ion. So the nucleotides of the DNA is different from the nucleotides of the RNA in such a way that the sugar which is present in the DNA is deoxyribonucleic acid, and the 5-membered sugar which is present in the RNA is ribose. So in this way, this is another difference between the DNA and RNA. Another difference between the DNA and RNA, you can see in this diagram that here these bases which are present, these are the names of the aromatic bases which are present in the DNA. A stands for adenine. G stands for guanine. G stands for guanine. C stands for cytosine. And T stands for thiamine. Thiamine. Thymine. Thiamine. So these four aromatic amine bases are present in the DNA: adenine, guanine, cytosine. In the RNA, adenine, guanine, cytosines are present. But the difference is between the fourth type of the aromatic amine bases. Here thymine is present in the DNA, while U is the abbreviation, or it stands for uracil. Uracil is present in RNA, while what is present, thymine is present in DNA. So this is another difference which you can see how the RNA and DNA are different from one another. Here the structure of thymine is given. Thymine is found only in DNA, and uracil is found in the RNA only. Uracil is found in the, this is the structure of the uracil. Uracil is found in RNA. Thymine is found in the DNA, while cytosine, this is the structure of the cytosine. Cytosine is found in the DNA and RNA. Adenine and guanine are also found in the adenine, guanine and cytosine. These are found in DNA and RNA. So this is another important difference between the two types of the nucleic acids. Now, nucleosides are nucleotides. Second topic is nucleosides and nucleotides. Now, if you are doing the hydrolysis, controlled hydrolysis of nucleic acid, then it produces the three components. Number one, heterocyclic aromatic amine bases. Number two, monosaccharide D-ribose or 2-deoxy-D-ribose. And third is the phosphate ion. Now the nucleotides is a compound containing what? Nucleotide is a compound containing D-ribose or 2-deoxy-D-ribose bonded to the heterocyclic aromatic amine bases bonded to heterocyclic aromatic amine bases. Here this one you can see the structure. A nucleotide is a compound containing D-ribose or 2-deoxy-D-ribose bonded to heterocyclic aromatic amine bases by beta N glycosidic bond, beta N glycosidic bond, whereas that of DNA is in the nucleotides. You have seen the components. The monosaccharide component of DNA is 2-deoxy-D-ribose. DNA is 2-deoxy-D-ribose, while that of RNA is D-ribose. Now, 5 these are the 5, 1, 2, 3, 4 and 5 these are the 5 aromatic amine bases which are found in the nucleic acid or the nucleosides and nucleotides. In the nucleosides and nucleotides, in the nucleosides, what are present in the nucleosides, aromatic amine bases derived from the purine or pyrimidines are present, and the second component is the D-ribose or deoxy-D-ribose. These two are present in the nucleosides. In the nucleoside, two components are present: aromatic amine bases derived from purine and pyrimidine, and deoxy-D-ribose or D-ribose. These are present in the nucleosides. And in the nucleotides, 3 components are present. One is aromatic amine bases derived from purine and pyrimidine, and sugar component which is D-ribose or deoxy-D-ribose, and third component is the phosphate ion which is present in the nucleotides and it is not present in the nucleosides. The aromatic amines, 5 aromatic amines are present in the nucleic acids. These are given the name accordingly. This is number 1, number 4, number 5. This is adenine. This is the structure of adenine. Here, this is the structure of purine. The structure of purine, and counting is done in this way. This is number 1, this is number 2, this is number 3, number 4, number 5, number 6. This is counterclockwise. Counting is done here, and then this is number 7, this is 8, this is 9. This nitrogen is 9. So this is what? This is purine, the structure of purine. This is the structure of pyrimidine. Again here counting is done in this way: number 1, 2, 3, 4, 5 and 6. The 3 aromatic amines which are derived from the pyrimidine, these are called as pyrimidine bases. Here you can see this skeleton structure of basic structure of the pyrimidine is present here, but the difference is here one carbonyl group is present, here another carbonyl group is present, and here NH is present, and with the corresponding change of the position of the double bond are removed. This is called as uracil. This is called as uracil. This is called as cytosine. Uracil is represented by U. This is cytosine, it is represented by C. Difference you can see between the uracil and this one is C double bond O, C double bond O is present. Here C double bond O is present, here NH2 group is present. This position, these two are different, and this is called as cytosine. And in the thymine, what is the difference? It has 2 carbonyl group similar to that of uracil. C double bond O is present here, C double bond O is present here. Difference is here methyl group is attached at this position. This is called as thymine. These 3 bases, these 3 aromatic bases are called as pyrimidine bases because they are derived from pyrimidine. These 2 bases, this is called as adenine, or it is represented by A, and guanine, it is represented by G. These 2 bases are called as purine bases because these 2 are derived from the purine. So one component of the nucleosides and one component of the nucleotides is present as pyrimidine or purine bases. Second component is, this is the open chain structure of the D-ribose. OH is present here on this side, CHO, CH2OH. This is open chain structure of D-ribose. Open chain structure of the D-ribose. Number 1, number 2, number 3, number 4, number 5. 5 carbons are present, OH are present on right side. This is called as D-ribose. At position number 1, what is present? CHO. This is called as, this is number 1, this is number 2. At 2 position, instead of H, there is present, instead of OH, here it is present H. This is called as 2-deoxy D-ribose. This is, these 2 are, this is the D-ribose, this is 2-deoxy D-ribose, open chain structure. This is ring structure of the 2-deoxy. This is 2 position, H is present here, 2 position, OH is present. This is deoxy D-ribose. Sorry, this is D-ribose. 2, here no mention of the 2 because deoxy is present here. This is D-ribose, cyclic structure of D-ribose. This is the cyclic structure of deoxy 2-deoxy D-ribose. 2 position, instead of OH, here H is present. So first component is pyrimidine bases, And second component of nucleoside or nucleotide is the sugar or D-ribose or deoxyribose. One component, this is the D-ribose, this is 2-deoxy-D-ribose. This is D-ribose cyclic structure. This is cyclic structure of 2-deoxy-D-ribose is present. So these two components are present in the in the nucleosides and in the nucleotides. And you have seen that here nucleotide consists of three parts. This is aromatic amine base, sugar component, and phosphatine or phosphoric acid component. Nucleoside consisting of only two components. This is cytosine, aromatic amine bases. This is a sugar. So the difference between the nucleotides and nucleoside is nucleotide is consisting of the aromatic amine bases and D-ribose. Those two components are present here. One is cytosine, which is the aromatic amine bases. This is what sugar is present, sugar component is present. This is what phosphate ion is present, which is not present in the nucleosides. So you can see the difference that nucleotide is consisting of three parts, while nucleoside is consisting of only two parts. No phosphate ion is present here. So keep this difference in your mind. This part is called as cytosine plus sugar part. Collectively it is called as cytidine. Cytosine is the aromatic base derived from pyrimidine base. It is a pyrimidine base. Cytosine and the D-ribose, this is called as cytidine. Its name is cytidine. Cytosine plus D-ribose sugar collectively is called as cytidine. And cytosine and 2-deoxy-D-ribose is called as deoxycytidine. Deoxycytidine. While the name of the nucleotide has three parts, while nucleoside has only two parts, aromatic amine bases and sugar. Nucleotide is consisting of aromatic amine bases, sugar, and phosphatine or phosphoric acid. How the name is given to the nucleotides. You have seen how the name is given to the nucleoside. This is called as cytidine when D-ribose and cytosine is present. And at two position, if hydrogen is present instead of OH, it is called as deoxycytidine. Now, how the name is given to the nucleotides? if this is cytosine, which is the pyrimidine bases, then sugar, then phosphate ion. This is called as cytidylate. So you have seen that different components of the nucleotides and the nucleosides. Here nitrogen bases which are present and how they will be named. If adenine is present as aromatic base, then the nucleoside is called as adenosine. And in the nucleoside, what is present with the adenine? With the ribose. It is called as adenosine. And when with the adenosine, what is present? Phosphate group is present, then it is called as adenylate. Aromatic amine is guanine. When it is present in the nucleoside, it is called as guanosine. And when phosphate group is present with the guanosine, it is called as guanylate. Cytosine is the pyrimidine base, and as a nucleoside is called as cytidine, and as a nucleotide it is called as cytidylate. Thymine is the type of the aromatic amine bases. It is also pyrimidine bases because it is derived from the pyrimidine. As a nucleoside, it is called as thymidine. As a nucleotide, it is called as thymidylate. Uracil is what type of the aromatic bases? It is a pyrimidine base, and as a nucleoside it is called as uridine, and as a nucleotide it is called as uridylate. So how you can give the name to the nucleosides and nucleotides? Naming of the nucleoside, here you can see this portion is what? Guanine. This is guanine. Guanine is attached from which carbon? This is glycosidic bond with the N9 nitrogen of the purine bases. With the purine bases, it is the linkage is glycosidic bond between the enameric or carbon number 1 of the D-ribose or deoxyribose to the N9 bond. While in case of purine bases, N9 nitrogen is attached with glycosidic bond with the D-ribose or deoxyribose. While in case of the pyrimidine base, N1 of nitrogen of the pyrimidine base will be attached with the glycosidic bond with the D-ribose or deoxyribose. And when purine base is present, N9 and glycosidic bond with the anomeric carbon will be formed. So what is this nucleoside? Nucleoside has two components, aromatic amine bases and the ribose or ribose sugar. So this is called as guanosine. These two collectively are called as guanosine or add-methylated guanosine, add-methylated 2-deoxy guanosine. ये मुख्तलिफ नाम आप इसको दे सकते हैं. What type of the linkage is present? C1 of the ribose and N9 of the purine bases will give the name to the aromatic bases and the nucleosides. D-ribose or ribose and aromatic amine bases will give you nucleosides. Now here linkage is C1N9, linkage C1N9 linkage is present in the purine bases. This is guanosine. This part is called as guanosine. This part is called as 2-deoxy D-ribose, 2-deoxy D-ribose. So it is called as add-methyl guanosine, add-methyl 2-deoxy guanosine. At this position, instead of OH, H is present. How the names are given to the nucleotides, and keep it in your mind that nucleotide is composed of, number one part is aromatic amines, it may be purine or pyrimidine bases. Number two, the sugar or carbohydrate part, which is D-ribose or deoxy D-ribose. And third is the phosphate ion, which is derived from the phosphoric acid. Three components are present in the nucleotides. Only two components are present. This part is present in the nucleoside. These three parts, when are present, these are present in the nucleotides. How the nucleotides are given the name? This part is called as guanosine, and this is called as guan, G-U-A-N. Osine is replaced with violet. Guanosine was present, the name for this part of the as a nucleoside. With this, it is called as guanosine, or this part is guanosine and it is modified into guanylate, or it is called as guanosine 5 dash. This is the 5 dash monophosphate. Guanosine 5 prime monophosphate. Another is called as adenosine. Adenosine, if here instead of guanine, adenosine is present. You can see the structure we have already studied. So it will be, if adenine is present, then this nucleoside portion will be called as instead of guanosine, it is called as adenosine. It is called as adenosine, and this will be adenosine 5 prime. If one phosphate ion is present, phosphate will be PO4 with -2 negative. PO4 -2 is phosphate ion is attached here. So that will be adenine, D-ribose, phosphoric acid. These three components are present, and it is called as adenosine 5' monophosphate, or it is called as AMP. And if here instead of one component, two components are present, instead of this two components are present, then look at the structure of the P here. This is the PO4. What type of linkage is present? N1, C1 and N5. This is purine basis में कौन सा linkage होता है? N9, C1, N9. और pyrimidine basis में कौन सा linkage होता है? इधर तो C1 ही होगी, उसमें क्या होगा? N1 वहाँ पे होगा, किस में? pyrimidine में. अच्छा, हम इसकी बात कर रहे हैं. ये पार्ट क्या होगा? एडेनिन था. ये पार्ट क्या है? डी राइबोज है. ये पार्ट क्या है? ये फास्फोरिक एसिड का पार्ट है. इसको आप नाम कैसे देते हैं? इसको AMP कहते हैं. AMP एडिनोसिन 5' मोनोफॉस्फेट. AMP. फिर क्या होता है? ADP एडिनोसिन 5' डाईफॉस्फेट. और इसके साथ एक और क्या होगा? PO. जिस तरह ये यहाँ तक हो, ये तो ये क्या होगा? डाईफॉस्फेट होगा. ये एडिनोसिन, ये क्या है? 5' एडिनोसिन 5' डाईसल्फेट. यहाँ तक हो तो फिर ये क्या होगा? डाईसल्फेट होगा. और ये P कितने हो गए? 3 हो गए, तो ट्राईफॉस्फेट. ATP एडिनोसिन 5' ट्राईफॉस्फेट. Adenosine 5' triphosphate. So this is the structure of ATP. This is AMP, adenosine monophosphate. This is ATP, adenosine triphosphate. And what is ADP, adenosine? This up to this structure here, O negative is present. This portion should not be present if it is not present. Here OH or O negative is present, so that will be adenosine diphosphate. Adenosine diphosphate. I think you will be able to understand clearly what is ADP, what is AMP, ADP, ATP, what are nucleotides, what are nucleosides, what are the nucleic acid, and what are the composition, how they can be given the name. I think you will be able to understand more clearly that what are the nucleic acids. Okay, then Allah Hafiz. इंशाल्लाह नेक्स्ट टाइम हम कुछ और चीज़ें इसी के साथ पढ़ेंगे, न्यूक्लिक एसिड के साथ. तो हाफिज़.